Welcome, everyone. Thank you for joining us at this trainee oral presentation session. My name is Jorge Castillo. I'm a graduate student at the University of Toronto, and I'm conducting research on ex vivo organ perfusion in the laboratory of Dr. Ian Rogers at the Mount Sinai Hospital in Toronto. And this year, I had the privilege of being part of the planning committee for this meeting. Francis.
So hello everyone, I'm Francis Migneault. I'm a research associate at the CHUM in Montreal, and I'm working in Marie-Josée Hébert's lab for quite a few years now, and I'm also the co-lead of the CDTRP education and career platform since 2018. So just before we start this session, I would like to share a few words with you. So in memory of Leon Timmons, who passed away on October 9th, 2023, at the age of 62, we pay tribute to a remarkable transplant nephrologist and researcher. Leon's immense contributions to the field and her unwavering dedication to the transplant community left a permanent mark on all those who had the privilege of knowing her. Leanne was instrumental in setting up the education and training platform. Under her leadership, the training platform fostered the creation of a national collaborative research network, bringing together trainees and early career researchers in a common quest for knowledge and innovation. Beyond her professional achievements, Leanne was a true advocate for building a sense of community within the donation and transplantation community. She understood the power of connection and worked tirelessly to create an inclusive and supporting environment for all. We will long remember Leanne's commitment to improving the lives of others, working tirelessly to advance the field of transplant medicine and improve patient care. Thank you.
Thanks, Francis. I will now invite Chelsea Lanos to come to the stage to talk about her project, Organ Donation After Out-of-Hospital Cardiac Arrest in Canada, a missed opportunity for UDCD. Chelsea is an Ontario-based advanced care and community paramedic, educator, and researcher. She's an adjunct lecturer at Monash University in Australia and holds a Master's of Science in Critical Care with a dissertation focused on organ donation after out-of-hospital cardiac arrest. Her research interests and developing program of study include looking at opportunities for donation from the out-of-hospital environment and the role of paramedics in donation pathways. So please welcome Chelsea.
Thank you so much, and thank you everybody for being here. Now, I'm sure some of you are wondering what a paramedic is doing at CDTRP and what a paramedic is going to teach you about donation and transplant. And when I think about this, I think about something that Key said yesterday in the first session, and she said something that really resonated with me. She said that we all come from backgrounds in which we are really comfortable. We all come from areas of practice in which we're very comfortable. And the key when we talk about organ donation is to figure out how to articulate and translate the knowledge that we bring from our particular environments to try to solve this puzzle. So I'm going to do just that. Like I said, I'm not here to teach you about donation. I'm not a transplant specialist. I'm not an expert in transplant and donation by any means, but I do come from a clinical background, and I can tell you from my experience that there is a lot of potential in the world that I work in and practice in. And so that's us right there at the bottom. We're a little piece of this puzzle, and I'm going to spend the next 10 minutes trying to convince you that paramedics might be a contributor to the answer to the problem that we are all collectively trying to solve. So I have no disclosures. A big thanks to my research team, and my affiliations are listed on the right. So to state the obvious, there's a worldwide shortage of organs, worldwide, and this has caused a rise in demand in those organs. And what that has, led us to is the exploration of alternative and somewhat unconventional organ donation pathways. And one of these pathways is the process of uncontrolled donation after cardiac death, or UDCD. And specifically, this pathway is quite viable in other countries internationally. And we wanted to know if this could be an effective solution to solving some of these problems that we see in Canada. So in other places worldwide, they're actually looking at that out-of-hospital cardiac arrest population and trying to see if we can identify potential donors within that population. So to give you a little bit of a background, there's about 40,000 out-of-hospital cardiac arrests in Canada, and when tended to by paramedics and treated by paramedics, so an attempted resuscitation by paramedics in that pre-hospital environment, only about 10% of those survive. So if you do some really quick math on that, there's about 36,000 patients that are not routinely considered for donation. And I can tell you from my clinical experience that a lot of these patients are actually left at home. So we resuscitate somebody at home, and when that resuscitation is unsuccessful, we don't often convey that person to the emergency department, therefore missing that opportunity in a lot of these cases. And of course, to contrast that, there's about 3,700 patients on wait lists, and we wanted to know out of that, 36,000 or so cohort of patients, how many patients could be eligible to solve this problem. So that's what we did. We decided to estimate the Canadian potential for, donors in this pre-hospital cohort, and we did that doing a 3-year cohort study using the CanRock Registry, and the CanRock Registry is the Canadian Resuscitation's Outcome Consortium, and we applied 5 UDCD criteria that's used in practice internationally to kind of see who out of that cohort could possibly be a donor. And what that did was create a range of potential from, best case to worst case scenario using the different criteria. So we included all non-traumatic adult OHCA cases from the CanRock Registry within that 3-year cohort that were treated by paramedics. And once we applied our exclusion criteria, we ended up with a study population of just over 9,000 patients. And we applied those 5 criteria for UDCD to that population. So you can see, I know this is a bit of a busy slide, so I apologize for that. There's just no real way, good way to do that. So what we did, and what's important to note here, is that out of these 5 criteria, and one was informed by subject matter experts, and what's really important, and I want you to pay attention to, is that we divided this by case characteristics. So that included things like age, whether or not the out-of-hospital cardiac arrest was witnessed, and then time to CPR, or whether or not there was bystander CPR. So we did that first to generate a best case scenario. And secondary to that, from that number, which I'll show you in a minute, we applied comorbidity data. And what's really important here to note is that paramedics, I can tell you firsthand, are really bad at documentation. We do not reliably capture comorbidities, especially in the context of running a resuscitation, in an acute crisis in somebody's house. So what we did instead was by proxy, we applied population estimates to our cohort, and we did that for every single one. So for example, we went to Health Canada or StatsCan, and we pulled stats for liver disease in Canada, for example, and we applied that to our study population, and we excluded those patients. And one thing too that is important to note is that we didn't account for patients with multiple comorbidities. So, if there's a person that had renal disease and hypertension, for example, and maybe was HIV positive, we would have taken 3 people out of our cohort, when really in real life, that just would have been 1. So the second thing that we did is we applied our second case, which was, after applying the second case, so the comorbidity data, we ended up with, our best case scenario, which was 3,700 patients. And then our worst case scenario after those applications, my apologies, was 830. So we ended up with that range of potential here, which I can show you here. So ranging from 830 potential donors from our cohort to just over 3,000 or 3,700 patients. And then the second thing that we did is we used an average of 2.1 organs per donor and rounded that down to 2 for real world application. And we ended up with twice the amount of potential organs that we could, potentially obtain from our study cohort. So if you contrast that to the numbers that you saw on the wait list, these are very powerful numbers. So a quick recap here at the numbers. So again, 40,000 out-of-hospital cardiac arrests every year, and when treated by paramedics, only about 10% survive, leaving behind a considerable population that we don't routinely consider at the moment. And then out of that population, we generate that range of potential between 830 to just over 3,700 patients. Now, in theory, that sounds great. It sounds really exciting, but the thing that we need to, to really put emphasis on is implementing these programs would be really, really challenging for many reasons. Paramedic education and scope of practice is one of those things. We need to talk about medical ethics. We need to talk about things like logistics, proximity to transplant centers and that kind of thing, as well as, several other factors. So all things to consider. Oops, my apologies. And one of the things to kind of alleviate that we need to think about is maybe considering other options for donation or for transplant. So I think, Sunny, you said, organs, are really sexy, but tissue is really expensive. So maybe we need to focus, our efforts on tissue donation from this population, for example, which would be a little bit less ethically challenging, logistically challenging. We're not operating within those time constraints. Another potential opportunity for paramedics is the involvement in NPOD after MAID in the community, which we are involved in already, and then maybe we can consider UDCD following that. So of course, our study had some limitations inherent to just being a retrospective study and using a registry. Even though our intention was to have a pan-Canadian representation of out-of-hospital cardiac arrest, we were limited by the data in the registry at that time, which was really only reflective of 3 provinces. And then, of course, we only considered adult non-traumatic OHCA, so we didn't look at trauma or any other special populations, including pediatrics. And then, of course, we added comorbidity estimates, which, as a proxy to real data seems good, but I think it would be, ideal to triangulate that data to be a little bit more accurate. So as a summary, there's an organ shortage and there's a large OHCA population that we're not routinely considering for any form of donation, whether that be tissue or UDCD and so on and so forth. And there is immense potential for, those pathways in Canada based on the data that we have. And of course, there is a role for paramedics to support those pathways, and that would lead to things like systemic change. So a look ahead for us as a research team, there is a long road ahead. There's a lot that we want to look at, but a front of mind for us is really just doing an evaluation of what's already out there. So we just put in a grant for a scoping review to see which countries are doing this and how they're doing it and how they overcame all of these barriers. We want to look at subpopulations. We want to look at quantifying, the other populations that we spoke about. We want to look at economics, the legislation surrounding and creating barriers for us, and of course the ethics around such programs, and then of course defining pre-hospital pathways for paramedics. And I'm happy to take any questions. Thank you for your time.
As well. I guess one of my questions is a lot of the countries that historically did a lot of uncontrolled DCD, specifically Spain, have been actually moving away from it over the last few years with instead considerations of ECMO CPR, which is a similar cannulation, but with the goal of attempting at least to save a life. Those patients might go on to become either NDD or DCD donors. And I guess the question is, have you considered, in discussing this, considerations for eCPR with the potential implications for donation afterwards, as opposed to uncontrolled DCD, and what those discussions and decisions were?
Turned off? Oh, perfect. Thank you, Matt, for your question. So truthfully, we didn't look at any of that. I know that some studies that we did find and we did look at within the literature did speak to that. Specifically, the study that we actually modeled our study, which was by Ho et al. In 2021, that's something that they accounted for. But as this was, my dissertation work, we didn't get into those things, but certainly something to consider here. So thank you for bringing that up.
This may be a dumb question, but what happens now? It does. No one in a hospital. So you arrive there, the paramedics arrive there, and what happens? Isn't there already a process where somebody, a coordinator or something, intercepts that whatever? No? Or
Yeah, so that's a great question. In theory, if patients are conveyed to the emergency department, there should be a referral pathway initiated in those emergency departments. Just based on, it's not something that I'm entirely familiar with, but based on my conversations with those that are, I have been told that our conveyance rates from the hospital are actually quite poor. But I think that the take-home message from this presentation specifically is all of the patients that we're not conveying to the emergency department. So we're initiating resuscitation, and when those attempts are unsuccessful, we're not actually transporting these patients, we're leaving them at home. I hope that answers your question. Right. Thank you so much.
Thanks, Chelsea. That was very, very interesting. Now inviting Shiyuan Bian to the stage to talk about his project on investigating the effect of mechanical ventilation on mouse lung scaffolds using a negative pressure wet ventilation system. Shiyuan is a third-year PhD candidate and then in the Department of Laboratory Medicine Pathobiology at the University of Toronto. In 2021, Shiyuan completed a Bachelor of Applied Science in Engineering Science with honors at the University of Toronto. His research focuses on the interplay between physical and mechanical cues on lung scaffolds and epithelial cells. So welcome.
Thank you, everyone. So my presentation now will get like a little bit technical. My presentation today is investigating the effect of mechanical ventilation on mouse lung scaffolds using a negative pressure wet ventilation system. So here I present to you a current data from Health Canada on the transplant stats on the year 2022. So you can see that this year, around 20% of the critical need patients die while waiting for a lung transplant in Ontario and Canada. So, okay, cool. Okay, thank you. It's okay. Yeah, sorry for the interruption. So this shows there's an increasing need for an alternative source for grant and transplantation. So an alternative of using donor lung to instead of like getting it from human is we can do recellularization of bioengineered lungs. So what that means is that we take lung from large animals like pigs. After that, we remove all the previous cellular content, plant it in with human cells, and then let it repopulate the lung and then transplant it back to the patient. And this serves as an alternative way of developing use functional lung for transplant. However, current systems that we developed to react to recellularize lungs have a sort of comeback shorts. And most current platforms use a perfusion-only system that does not take into the account of the dynamic environment in the lung, because lung is currently being ventilated to allow the exchange of air. And also the ventilation rate that for those systems that incorporate ventilation are very low. So for example, this paper over here, it shows that around 1 breath per 2 minutes, which is much, much lower than our normal breathing rate. So to overcome this problem of too low ventilation, we developed a novel designed negative pressure wet ventilation system. It will allow us to have a wider range of tidal volume up to 2.0 microliters, a longer system running time, which is 5 days, and a higher ventilation rate, that's the most important part, up to 32 breaths per minute. That's 64 times better than the other systems. So to use this new system, we can use it to examine the effect of the cyclic stretch on lung scaffolds, because we want to make sure that the scaffolds are not damaged by the ventilation, so the lung becomes unfunctional, and also to examine how the cells perform in the cyclic stretch condition. So we designed some experimental groups to test the effect of cyclic stretch on the lung using our system. So for the controls, we have the native, the decellularized lung only, and the decellularized with perfusion. Those are the current protocols. And then using our ventilation system, we have 3 groups. One is like a normal ventilation with a low breathing rate. One is a normal tidal, the other is a normal tidal volume with a higher rate. And the last one is that we increase the tidal volume to a damaging level to see how a damaging ventilation could do to the lung scaffold and the cells inside. So first, we remove all the cellular content inside the lung. The result shows that 99% of all cellular content was removed. And then using the mouse model, we put it in the negative ventilation system and then start ventilating. Then we perform the mechanical testing using a uniaxial tensile stress testing system to see how the stiffness of the lung changes with ventilation. So for the setup, we get the left lobe of the lung, and we cut of the mouse lung, and we cut it to a 5 millimeter times 2 times 1 millimeter long strip, and we put it on a platform called biaxial tensile testing system, where there are 2 hooks on both ends, and they will hook up the lung and stretch out uniaxially to test when the lung breaks. And during this time, we can get the stress and strain curve, which is a measurement of the stiffness of the lung. And here are the results. So sorry if the results are not clearly seen on the screen, but the native lung has the highest stiffness, and the overventilated lung, which is kind of act as a damaging control, has the lowest stiffness. But all the other lungs, including the decellularized lung and decellularized lung with a normal ventilation rate and higher ventilation rate, was in the middle, and they are not significantly different from each other. And this is the detailed stress and strain measurements. What we got is that at a strain rate of 0.8 and 1 kilopascal, strain of 1 kilopascal were all consistent except for the damaging control. So what this means is that with a normal rate of ventilation and a normal tidal volume, we don't change any significantly, like any mechanical factors that's affecting the lung, which shows that our ventilation system won't damage the lung scaffolds. And just a summary that this mechanical property of the moderate ventilation lung have no significant difference than a perfusion lung, meaning our system is okay with ventilating lungs without causing further damage. And next step is to examine how the cells react to this ventilation. And what we did was that we take our decellularized lung, and first we ventilate it and then cut into small pieces and then see cells on it to see the cells respond on the scaffold. Second is that we take the decellularized mouse lung, we see the cells in them first, and then we do the ventilation to see how the cells react to this whole lung recelled system. And after that, we examine the cell coverage rate, viability, and I'll first introduce you about how our whole lung recellularized it. So we take this A549 cell, which is a cell line currently used to mimic the alveolar epithelial cell, and we seed the cells in, let it culture for 18 hours inside the lung to let it sit. And after that, we perform 5 days of ventilation. And there's also 2 control groups where there's perfusion only and no ventilation and perfusion. And then we did the analysis. What we found was that the coverage rate for the cells that's in the ventilation group is much higher than the perfusion only and no perfusion, no ventilation group. This shows that the ventilation helps the cells spread out and survive. You can see in the image over here, like the perfusion only group, the cells are clustered together, but in the ventilation group, the cells are spreading out evenly into the alveolus. This is a much better looking recellularized lung than the perfusion only. And then to see cells on the scaffold after the ventilation, we use a technique called the precision cut lung slices, where we first inject agarose into the lung, and then we cut it using a small blade called microtome, and then we image the cells on the slice. And using the same A549 cell, we see that in the ventilation group compared to the control group, there's a higher coverage, higher viability, and even a higher proliferation rate than in the ventilation group compared to the no ventilation group. And then we move on to test how primary 82 cells, which is cells directly harvested from donors, act on the precision lung slices. And we also including a high rate ventilation group compared to our moderate ventilation group. And what we see is that both ventilation groups performed much better in improving the coverage rate and viability of the primary cells on our slices. So a summary of this finding is that we find both the cell line and the primary alveolar epithelial cells is improved by the ventilation in both proliferation rate, coverage, and viability. And we also found that a higher rate of ventilation seems also have a better performance than the moderate rate of ventilation. And the cell coverage for the whole lung is also a very promising result, showing that ventilation on the whole lung system is also improving cell viability. And I think for my study, the outcome will increase our understanding in the impact of mechanical ventilation on lung structure in both with and without the presence of cells. And this could lead to the regeneration of more functional bioengineered lungs for transplantation. And I want to acknowledge my supervisor, Dr. Gones Karubi, and Dr. Siba Haikal, and all the other members of my lab. And thank you for their support. And thank you for listening.
Recognizing that you've worked on lung architecture, how do you think this is translatable to some of the other organs, kidneys, livers, etc.?
Sorry. Yeah. Okay. So that's good because I think this is translatable because one issue that we are facing is that if we do those ex vivo studies on lung scaffolds, a lot of time is a lot of mechanical cues are not studied. For example, in lung we have ventilation, in kidney we have renal flow that has act as a shear stress. That's also a mechanical factor. And to study like how they affect the structure will let us know like what kind of parameters we can use to culture the cells to let them proliferate and grow normally, but not damaging the structure, because decellularized scaffolds not doesn't matter if it's lung, liver, or kidney, will have a decreased structural stiffness because the cellular content is also adding to make the structure very strong. If you remove those cellular contents, it could make the structure more fragile. And if you apply like a normal physical cue, same magnitude as we experience in vivo, it might damage the structure. And if the structure is damaged, the skin is less likely to become functional. So that's why it's important to study how this mechanical cue affect the structure of decellularized lung or kidney or liver.
Thank you. Excellent work. And then maybe one more question, if nobody else has this. You're starting with a pig lung as your infrastructure, your scaffold. Pigs are only so big. If way down the line you're using the scaffold, will it grow as well once it's recellularized?
First of all, in my experience, we are using mouse lung, but if you are using, if you're using like
a pig in the picture.
Okay.
And I thought, could you move this to other scaffold?
For sure. Yeah. So just explain the current why we're using mouse. It's like it's small, it's easier to handle, and it's been studied thoroughly. And if we like want to use pig for example, so I would think that the scaffold, because the cells can also modify the ECM that they are living in. So, but I don't think that the lung will likely to grow with the patients like growing because they are not a human lung, they are not isogenic. So but I do think that this technique can be used for like adults when they are not growing anymore. And we can also, because we are taking large animals, there's also different size of large animals we can use. So to fit different size of human. So yeah.
Thank you. Nice work. Nicely answered.
Thank you.
So before moving to the next speaker, I have a question regarding your model. So you saw some assays to assess the viability of the alveolar cells. Did you or are you planning to study the function, the ability of the lungs to resolve edema in the future, like wet to dry ratio or?
Yeah, so that's a good question. So currently we haven't moved, we are like a little bit far from that step. We first want to make sure that the cells are first, they are surviving, and two, they are spreading, third, they are proliferating inside the lung. That's the first thing we want to do. I think after we can confirm all three with a pretty good magnitude, we can then go ahead and study how the cells respond to, for example, edema, or how, like, whether there is edema inside the lung. But first, we need to get a like towards a functional lung, and then that's the next step for it.
Thank you.
Thank you. Thank you.
So I will now invite Aisha Adil to the stage to talk about her project ex vivo perfusion, de and recellularization of rat hind limbs for vascular composite of allotransplantation. Aisha is a master graduate through Institute of Medical Science at University of Toronto under the supervision of Dr. Siba Ekal and Dr. Golnaz Karoubi. Aisha previously obtained an honors Bachelor of Science at the University of Toronto, specializing in health and disease before proceeding to graduate studies. Our research focuses on tissue engineering, composite, and rat hind limbs using tissue de and recellularization for vascular composite allotransplantation.
Thank you for the introduction. I'll reintroduce myself. My name is Aisha. I just finished my MSc with Dr. Siba Ekal, and I'm really excited to present our work on the ex vivo perfusion, decellularization, and recellularization of rat hind limbs for vascular composite allotransplantation. Vascular composite allotransplantation, or VCA as we'll refer to it, is an emerging reconstructive surgical option for patients who've undergone severe tissue loss and musculoskeletal damage. This can include patients who've had traumatic injuries, severe burns, or tumor removal surgeries, for example. And current therapeutic options include autologous reconstruction, where you use your own body to repair the injured area. And while this is the current standard, it's pretty limiting because there's limited donor tissue availability, donor site morbidity, need for multiple surgical interventions, and so forth. And VCA entails the transplantation of multiple tissues, such as skin, vessel, nerve, muscle, and bone, all together as a composite subunit from donor to recipient. And while this is very promising, immunosuppression regimens are very stringent and lifelong that can have severe side effects for patients. Some examples include malignancies, opportunistic infections, to name. And so tissue engineering approaches represent a potential solution. And a particular approach for tissue engineering is called decellularization and recellularization. And to walk through what that means, we first started off with obtaining a tissue from a donor that is then subjected to decellularization. Decellularization entails the removal of cellular and nuclear content while maintaining the extracellular matrix of the scaffold, or in other words, the 3D structure of the tissue. And what this does is it creates an acellular scaffold that can then be subjected to recellularization, which is the repopulation with recipient-specific cells, resulting in a transplantable tissue in the recipient. And in considering this technique for VCA, we have previously reviewed and summarized some of the different factors that are required for employing this technique for VCA, and we'll talk about each of these within this project. Now, taking a step back and looking at where this started from, decellularization and recellularization really began with work in the whole organs, such as the kidney, liver, and lung, to name a few examples. And then it was employed in VCA across various different tissue types, as well as across different animal tissues and human. However, there's still some pre-existing challenges, particularly with regenerating composite tissues that are on the whole more challenging. And this is due to a few factors. The first, that there's multiple tissue compartments that each have tailored needs. There's many different cell types that are required that are dependent on the type of tissue to be regenerated. And lastly, there's a longstanding challenge of regenerating function in tissues such as the muscle, the nerve, and the bone. So this brings us to the objective and goal of our project, which was to decellularize and recellularize composite rat hind limbs to establish a proof of concept small tissue engineering model for VCA. We started off with establishing first an ex vivo bioreactor system for decellularization. The system employs the use of a peristaltic pump that continuously perfuses detergent into the cannulated femoral artery, which is pictured here in the bottom right. The hind limb is also simultaneously submerged in detergent. And what this design of the bioreactor does is it enables us to have a closed system circuit, allowing a single pass perfusion of detergent through the hind limb. Using the system, we were able to successfully decellularize a hind limb using a low concentration of 0.25% SDS or sodium dodecyl sulfate, an ionic detergent that's commonly used in decellularization. And this was achieved in a period of 5 days. And we indicate decellularization using gross morphology, which is given by this white translucent-like appearance of the tissue. Next, we assessed each of the tissue compartments histologically. So starting off with the large vessels, the femoral vein and the femoral artery, we noticed that after decellularization, the structure of the vessels was maintained, but nuclear content was removed. When looking at the nerve, we also saw that there was nuclear content removed, but the preservation of the tissue structure was preserved up to the innermost layer of the endoneurium. When looking at the bone, we saw that the cells were removed from the endosteum, the inner layer, the bone matrix, as well as the periosteum, the outer layer of the cells. After decellularization, the structure was maintained. When looking at the skin, the cell-rich layer, the epidermis, was removed after decellularization, while the dermis with the collagen fibers was maintained and cell nuclei were removed. And lastly, looking at the skeletal muscle shown in a cross-section view, we see that the myofibers are maintained after decellularization, but the nuclei that are otherwise located at the peripheries of these myofibers are removed. To corroborate our findings, we then performed DNA quantification, where we saw that after decellularization, DNA levels were reduced after decellularization. Given that we had a decellularized scaffold, we moved on to recellularization. And in doing an overview here, we used human umbilical vein endothelial cells, or HUVECs for short, and we also used rat myoblasts, or muscle progenitor cells. HUVECs, we took 20 million HUVECs that were perfused, that were suspended in cell suspension and perfused into the femoral artery. And at the same time, we took 20 million myoblasts in a cell suspension and injected them throughout the muscle of the hind limb. This decellularized hind limb was held in static culture for 2 hours, after which it was started in perfusion using our ex vivo bioreactor, and this perfusion was continued up to 7 days ex vivo. We then assessed these tissue compartments histologically, both the femoral vessels, the large vessels, small vessels, and the muscle. So starting off with the large vessels or the recellularized femorals, we noticed that after 7 days of ex vivo culturing, some cells could be observed in parts of the intimal layer, where we could see some clusters of cells and some flat cells. In additional biopsies, we saw that in select locations of the vessels, there were cells lining the intimal layer, though this was not consistent across the whole intimal layer. We confirmed this using DAPI staining to see the presence of these cells similar to the H&E, where cells were located in sparse locations of the femoral vessels. However, to truly discern whether these were HUVECs or not, we used CD31 expression. We saw that CD31's staining was positive where in these select locations where cells were observed, and in some cases CD31 was seen to be consistent in some locations of the intima. What was perhaps more interesting was that several small vessels that are located more distally in the hind limb showed presence of cells in a more consistent manner, where the lining in cells and engraftment was more consistent, as seen by both the H&E and DAPI staining. And in some cases, we saw that there were clusters of cells in the lumen of the small vessels. And following the same story of confirming whether these are HUVECs or not that we were observing, we noticed that there was a consistent lining of CD31 along the intimal layer of these small vessels in contrast to the femorals, indicating that there's a potential new endothelium formation. And in some cases, we also observed a cluster of cells. Now, as you recall, I also mentioned that we recellularized the muscle at the same time. And so when we looked at the areas of the injection locations, we noticed that there was aggregates of cells found at these respective injection sites where cells were aggregated together and the cell nuclei was densely stained by both H&E and DAPI staining. However, because we were seeing these clusters after 7 days of keeping the hind limb in culture, we questioned whether these cells were alive or not. We then proceeded to do a fluorescence detection for apoptosis using tunnel staining as evidenced by this red fluorescence marker in the middle panel and counterstain with DAPI to look at cell nuclei. And looking at the right 2 panels of the recellularized muscle, we noticed that many cells were in fact staying alive after 7 days of ex vivo culturing. And quantifying the number of apoptotic cells relative to the DAPI stained cells across the recellularization experiments, across 5 of these recellularization experiments, and approximately 39% of cells were found to be apoptotic, indicating that potentially 61% of cells are remaining alive. Next, we wanted to phenotype these cells. Given that many cells were alive, we looked at the expression for myosin heavy chain. This is a marker for mature muscle marker. Usually when myoblasts start to differentiate and fuse together, they start to express myosin heavy chain that indicates that they're moving to a myotube formation. So we saw that after recellularization in these clusters, some myosin heavy chain expression was found surrounding DAPI stained nuclei in close proximity with each other. And this is reminiscent of a nascent myotube formation, where myosin heavy chain is starting to be expressed across along with DAPI stained nuclei in close proximity with each other, but they're not at that mature myotube stage, which is characterized by an elongated myotube. To conclude, 0.25% SDS perfusion decellularization was successful in systemically decellularizing the rat hind limb. We saw this through the loss of nuclei as well as reduction in DNA content. We also established an ex vivo perfusion bioreactor system that was suitable also for decellularization as well as recellularization. And our recellularization work with HUVEX and myoblasts showed that we had cell presence not only after 7 days of ex vivo culturing, but we also saw that there was a new endothelium and nascent myotube formation after this period of time. Pointing to some exciting work with our future work is incorporating electrical stimulation with recellularization, and this is with the purpose of establishing its role in enhancing muscle cell differentiation to our more mature muscle fiber phenotype. And so we're really interested to establish the use of electrical stimulation, where we want to find a system through the use of carbon rod electrodes. And this was inspired by the picture shown here of the electrical stimulation chambers. And I wanted to attempt adapting this to the whole hind limb. So during the first feasibility study, where we looked at whether these carbon rod electrodes can stimulate a full hind limb, we saw that there was some contraction of the muscle in this ex vivo hind limb. But then we started to question, can we fully submerge this hind limb in culture media that we typically use for recellularization and still see stimulation being delivered? So we then proceeded to a deep dish Petri design where a limb was fully submerged in culture media used for recellularization. And we noticed that there was stimulation being delivered in this system, giving us inspiration of potentially applying this with our recellularization. So given that the first 2 pointers were covered, we looked at the feasibility of these carbon rod electrodes, and we saw the successful applicability in submersion. We then wanted to design an ex vivo bioreactor that incorporated stimulation, where we saw that contraction was observed. And even with an airtight seal lid, we could still deliver stimulation ex vivo. So while this future directions are very exciting, we have to take a step back and really look at what contributions have we really made to the field. And the first is that we've established a small tissue engineered model for VCA that can help us provide a foundational basis for defining and refining more of these recellularization strategies for composite tissues. And lastly, with our recellularization, we've also shown that there's nascent tissue regeneration in composite decellularized hind limbs, both in the case of seeing a new endothelium regeneration as well as nascent myotube formation. And these critical findings are working towards using tissue engineering strategies, which can advance us closer to using bioengineered tissues for VCA, and this can be a promising solution for implementing VCA clinically. And with that, I'll end with acknowledgements to my supervisor as well as our lab members and open up for questions. Thank you.
Good talk. Thank you very much for the talk. I have a question about the recellularization. So basically, so you put the cell back to the ECM, and then the question is, if you culture for 7 days, so not all the ECM will have the cells, right? So some area will have no cells be there. So will the ECM be degrade during that time? Like
will the
7
ECM
days?
what, sorry?
Degrade.
Degrade?
Yeah.
Well, I haven't really assessed the ECM afterwards. It's also possible that the cells we repopulate with, they could secrete certain factors that could promote ECM remodeling, and it has been shown in previous studies. So while I don't know if it will degrade or not, we would have to keep seeing if it's been maintained. The goal is that with the decellularization, the ECM is maintained to help support the cell engraftment and eventually the cell function.
Thank you.
Fantastic work, Aisha. I'm curious whether you think that incomplete endothelialization is due to too few cells, incomplete delivery, or something else?
That's a great question and something I've thought about a lot. So in terms of recellularization, getting to this number of 20 million cells was based on a previous metric of using 1 million cells per cubic centimeter. So we took the volume of the hindlimb to do this. And the fact that we were able to see consistent engraftment in small vessels, but not the larger, indicates that potentially we may need more cells to reach that larger surface area, because this volume that we had derived of the hindlimb, it's based on the whole hindlimb. It's not necessarily taking the volume and surface area of the actual vasculature, which could be very different. So it could be, yes, maybe we have too few cells. It could also be just how we've recellularized. We kept dynamic perfusion ongoing through the femoral artery, but maybe we want to do perfusion both through the arterial and the vein to see if we can allow like more perfusion and nutrients to access the vasculature. Though I will say that being able to reach the distal vasculature and seeing consistent engraftment is a bit of a push in terms of the frontiers of recellularization, given that previous studies using not just the rat or mice, but also in pig and some human cadaver tissues, we weren't even able to reach that extent. So I think now we have a basis that maybe this metric that we've established is not the one we want to go with. Maybe we want to consider more advanced ways of measuring that surface area and volume and trying to derive more cell numbers or a metric to repopulate with.
I would just like to jump in with questions from Anna regarding this the endothelialization. So could it be possible that the cell type you use could be I mean, UVEC cells are a great cell model for endothelial cells, but it's still a vein phenotype. So
yeah,
could have been another cell type that could have been more appropriate.
Yeah, of course. So HUVECs, we chose them mainly because these are the standard proof of concept that have been used. So they're very easy to work with, easy to grow, and we can establish that proof of concept approach, but they're terminally differentiated. They're also not tissue specific. So another cell population that would be very appealing is endothelial progenitor cells. Although they haven't been well defined in terms of their isolation properties. But if you do use them, you can actually differentiate them. They do have the capability of differentiating into tissue-specific endothelial cells. So I think that would be a very appealing avenue to go with and see if we could really get tissue-specific re-endothelialization.
Great,
Yep.
thank you.
Great.
So I'll now invite Javairia Rahim to the stage to talk about her project. Novel cell surface engineering of the endothelial glycocalyx with sialic acid containing polymers for the prevention of transplant rejection. So Ms. Rahim is a master's student in Dr. Jonathan Choi's lab at Simon Fraser University, studying the role of cell surface engineering in preventing transplant rejection. In 2021, she completed her bachelor's in biochemistry with a co-op distinction at the University of Victoria. She is a former acting director of events and communications for the Society of Women in Science and Technology.
Hello, thank you for joining me today. So as we know by now, hopefully, the transplantation procedure involves a donor organ being transplanted into a recipient, and that they are connected through blood vessels. And so these blood vessels are lined with cells called endothelial cells. And so these endothelial cells have a extracellular matrix called the glycocalyx that is made up of lipids, proteins, sugars, and is protective to endothelial cells. And so during the transplantation procedure, during the organ procurement and transplantation procedure, blood vessels become damaged due to mechanical and ischemic damage. This causes the shedding of the glycocalyx matrix, and that results in the loss of endothelial cell protection, the exposure of foreign antigens, as well as the release of damage-associated molecular patterns. And so these factors contribute towards the pro-inflammatory cascade that contributes towards transplant rejection. And so this cascade involves immune cell recruitment, the development of donor-specific antibodies, as well as a secretion of pro-inflammatory cytokines. And so current strategies that have been employed involve the use of immunosuppressive agents. And so although those help in providing some protection, immunosuppressive agents are associated with adverse effects such as increased susceptibility to infections. So to circumvent the use of broad-acting immunosuppressants, our lab, the Troy lab, as well as labs at UBC, the Kazakh-Dathu lab, as well as the Withers lab, came together to co-develop a cell surface engineering protocol that can be used during the organ transplantation procedure. And so what this protocol involves is the enzymatic adhesion of the sialic acid containing immunoregulatory molecule polymer that can be adhered to endothelial cells to provide protection during that transplantation procedure. And so this polymer is called LPG-SIA, and it has a sialic acid residue, a polymer scaffold, as well as a glutamine-rich tail. And so this enzymatic addition involves the glutamine portion on that tail to be covalently bound to lysine residues found on endothelial cells. And so this causes endothelial cells to be covered in the sialic acid-rich polymer. And so sialic acid is an immunoregulatory molecule that can bind to immune receptors. And so a family of receptors it can bind to on immune cells are called Siglecs. And so these this family of receptors, there are different ones found on different cells. And so upon binding to sialic acid, they can cause immune cell activation, meaning that immune cells are more prone to undergoing their effector mechanisms, or they can, upon binding to sialic acid, cause deactivation to those immune cells to prevent them from causing immune-mediated cytotoxicity. And so the way that sialic acid is presented on LPG-SIA is that it could bind to Siglec-7 or Siglec-9. And so these are two Siglecs that are immune inhibitory. And so to provide some background on some previous data, so we used a kidney ischemic model to determine whether LPG-SIA can prevent kidney injury. And in this model, what we do is we perfuse kidneys with a control solution or a solution with LPG-SIA plus the enzyme. And what we see here is that we have an increased amount of survival of these mice after transplantation with the LPG-SIA polymer, and we have a significant reduction in kidney cell injury when we modify cells with the kidney with LPG-SIA, indicating that LPG-SIA can protect against ischemic kidney damage. In addition, we perform mouse allografts. So we take a donor artery from a biopsy mouse, we perfuse it with a UW control solution, or we perfuse it with our LPG-SIA polymer solution, and we transplant that artery into a genetically complete different mouse. And so through this, we're able to score for acute and chronic rejection at 15 and 42 days. And what we see here is after modification with LPG-SIA, we see a significant reduction in both acute and chronic rejection. And so to understand how endothelial cells are being impacted in this model, especially the acute model, I labeled these arteries with a marker called CD31. And so CD31 is found on endothelial cells specifically. So in xenografts, we see a complete lining of the lumen as denoted by the red line. And so we have an intact luminal layer. In the UW control, we see areas where we have the absence of CD31 staining. And so this indicates to us that there has been endothelial cell injury, which prevents them from expressing the CD31 molecule. And these areas are significantly reduced when we modify the arteries with LPG-SIA, indicating that LPG-SIA modification of mouse allografts prevents endothelial cell injury. But how exactly is this happening? And to understand that, I produced a in vitro cytotoxicity assay where I grew endothelial cells for 7 days. Then I stained them fluorescently with a fluorescent dye. I then modified the endothelial cells with our polymer or with a sialidase enzyme, which cleaves sialic acid. And then I incubated those with IL-2 activated PBMCs. And after this co-culture, I then measured the amount of fluorescence within the supernatant. And that is an indicator of endothelial cell death. And so what we see here from our results is that in 2 different donors in their unmodified control group, when we add IL-2 activated PBMCs, we have a lot of endothelial cell death. And that death is significantly reduced when we modify cells with LPG-SIA, indicating that LPG-SIA is protective towards endothelial cells and can deactivate PBMCs to prevent them from causing immune-mediated cytotoxicity. To understand whether it is specifically the sialic acid residue on endothelial cell, on LPG-SIA that is causing this immune cell deactivation, I modified these endothelial cells in a variety of ways. And these are flow cytometry plots looking at the amount of sialic acid. And so I modified cells with sialidase, so I cleaved sialic acid off of endothelial cells. I modified cells with LPG-SIA, and then I modified cells with LPG-SIA and then sialidase to determine whether it's specifically the sialic acid residue that is causing this effect. And what we see here is that when we modify cells with sialidase, we cleave off endogenous sialic acid, which is causing increased endothelial cell death, which shows that endogenous sialic acid is protective to endothelial cells. And as we know before, when we modify cells with LPG-SIA, we have an enhanced protection of our endothelial cells, and that protection was significantly lost when we first modified cells with LPG-SIA and then modified it with sialidase, indicating that it is specifically the sialic acid residue that is causing this effect. An immune cell population that can be involved in this immune-mediated cytotoxicity involves NK cells. And so I performed the same experiment, but with NK cells instead. And we saw similar results where when we modified endothelial cells with the sialidase, we saw a significant increase in endothelial cell death. And when we modify cells with LPG-SIA, we have a enhanced protection from NK cell mediated immune cytotoxicity. And so from these assays, I took NK cells from the supernatant and I performed flow cytometry to look at the externalization of CD107A. And so CD107A is found in vesicles that hold cytotoxic granules. And so the externalization of this molecule indicates the externalization of cytotoxic granules. What we see here is that there's some indication of increased expression of CD107A on NK cells when we modify cells with sialidase as compared to LPG-SIA. Due to high variability in the data, we weren't able to get a significance out of it, but with more replicates, hopefully we'll be able to resolve this issue. And so to understand how exactly sialic acid is causing this effect, I performed similar assays with neutralizing antibodies to Siglex. And what I noticed here and what we see here, I'll walk you through the graph. In our unmodified controls, we have a lot of endothelial cell death without LPG-SIA, and next to it is our antibodies to isotype, Siglec-7 as well as Siglec-9, and we see no non-specific endothelial cell death. Upon modification of our endothelial cells, as described before, we have enhanced protection, and we see no difference when we add our isotype control antibody. And once we add neutralizing antibodies to Siglec-7 or Siglec-9, we see some loss of that protection. And so it is only when we add both neutralizing antibodies to both Siglec-7 and Siglec-9 do we see a significant loss of that protection by LPG-SIA, indicating that LPG-SIA is actually binding to both Siglec-7 and Siglec-9 to cause that immune cell deactivation within this PBMC population to prevent immune-mediated cytotoxicity. And so in total, in mouse allografts, LPG-SIA modification prevents endothelial cell injury. It prevents, it deactivates PBMCs, it deactivates NK cells, and LPG-SIA binds to both Siglec-7 and Siglec-9 to deactivate PBMCs. And thank you.
So thank you for your presentation. I open the floor for the questions.
Very interesting work. I'm curious, if you were to look at the kind of traditional paradigm of antibody-mediated rejection, which is trying to mimic, the antibody that's bound to the endothelium activates NK or myeloid cells via Fc receptors. So where is the antibody here? And how is the NK cell mediating the killing?
So the way that I hypothesize that this is occurring in these cytotoxicity assays is through NKG2D receptors on NK cells binding to MICA, MICB ligands on the endothelial cells rather than through the antibody-mediated rejection model. Yeah.
I guess my question is just whether this is then translatable or relevant, although very interesting.
Yeah. So in terms of how this relates to transplantation specifically, although it doesn't really look at antibody-mediated rejection, it is testing for whether or not this this model allows us to look at whether we can prevent NK cell and T cell mediated cytotoxicity. And so far, like without antibody mediated rejection in the picture. So NK cell and T cell mediated, specifically T cell mediated cytotoxicity is what we see in acute arteritis. So in this model allows us to kind of probe at that specifically. Yeah.
I do have a question. This is very interesting. When I was wondering whether the sialic acid would have or whether all immune cells would have or would be responsive to the modification of the endothelial cells with sialic acid.
Yeah, so sialic acid is present on almost all cells, and so for this specific polymer, the sialic acid residue is presented in a very specific linkage that can bind very specifically to specific receptors. And that's kind of the idea behind it. And sialic acid all over cells are presented in a variety of ways. And so it is really dependent on the way that it's presented. So this sialic acid molecule, so far from what the data shows, that it can bind to Siglec-7 or Siglec-9 on a variety of immune cells, but more specifically NK cells and T cells and some monocytes as well.
Thank you.
All right.
We'll now invite our final speaker for this session. His name is Nicolas Bourgeois to the stage to talk about his project of virtual rehabilitation program in lung transplant candidates. Nicolas is a physiotherapist at the University of Montreal's Hospital Center and has worked in the lung transplant program for over 13 years. He completed his master's degree at the University of Montreal and is currently a PhD candidate in rehabilitation sciences at McGill University, which with a research focus on rehabilitation in lung transplant candidates.
So thank you. So thank you for this CDTRP. Allows me to talk about that today. So I'm going to switch a very different way of approach since the last few presentations. It's more a clinical approach for today going in. So, and this work, I want to start to say is coming from a CDTRP innovation grant that we received 2 years ago, which is good to present, and some funds from the CDTRP. So thanks for the CDTRP for that working on out. So first, the background, we know is lung transplantation candidates have they have dyspnea, they have decreased exercise capacity, decreased muscle strength, and support frailty regarding transplant. We had some association with knowing the literature between exercise capacity and mortality in these patients. And around 2 are against delisting for transplant with frailty and even disability with frailty too is association. An exercise consensus statement made by the Canadian Society of Transplantation and Can Restore talked a bit about the prehabilitation is recommended for these patients regarding that it was able to improve the physical condition and psychological to help before the surgery and have the goal to have a faster recovery after the transplant. It's not a goal. I don't have the time today about talking about prehabilitation. We had a little bit the glimpse of it previously today about what's rehab. So I'm not going to be that much definition of rehab because of time today. But it's quite a lot of topics we can discuss a lot of time about that. So regarding the number of RCT in exercise intervention in lung transplantation candidates, there's a very limited number in literature right now going in. Later on, there is a very quietly published in the last few months was a consensus statement about prehabilitation made by European Society of Organ Transplantation with solid organ transplant with all livers, kidneys, lungs, and all that. In that part consensus statement, they said that for lung transplantation candidates, it's unknown with the optimal delivery time. And also the exercise components regarding the rehab. So there's a need still there with the consensus statement made. And normally now in literature, what was published for past few years, all the intervention was mainly hospital-based or a little mix of hospital-based and home-based a bit more program. We know as part for transplant candidates is the same thing in Quebec for our patients. They're waiting home, which is quite not around the hospital. So they're way around all the provinces. So it's not maybe the best way to make them a bit more traveled. We know with lung transplantation candidates, they have a lot of oxygen normally with advanced disease. So make them move to go to the hospital. Maybe it's not the right maybe way of doing it with hospital base. And because of the pandemic, we had a big boom of telerehabilitation that went. So many programs of lung transplantation and a lot of solid organ transplant too, they mix and they change around center-based to more telerehabilitation. With the use of different technology tools that we know, there's still not that clear about the safety of the monitoring of these patients is going at home. There's a lot of studies going on that, but we know that telerehab and all the technology tools can help to and enhance the bit of communication between the healthcare providers and the patients. Again, increasing the uptake of the rehab and the adherence to the exercise at the same time. Regarding the effectiveness of telerehabilitation or like a home-based prehab in lung transplantation candidates particularly, there's very limited evidence there. There's 2 recent studies done. The latest study, they use it an app-based with cystic fibrosis patient going in with a 12 weeks size. Oh, okay. Why? Okay, it jumped out. I don't know why. And around the senior study, they use saw how it was 8 week. It was app-based around a bit more intervention, but they exclude some patients with pulmonary hypertension. So there's not that clear around. So it's really the app-based a bit more method. In our centers and working with these patients for many, many years now, we have the past using more given and exercise booklet to these patients, and we're not doing so much follow-ups with these patients. We published a retrospective study that we analyze what we've doing for a lot of years. And regarding the counseling that I did with patients when they come, I give them booklets, and after that, I was counseling when they come back to clinic at each 3 to 5 months around that. And we've seen in our study that the patients, many of the patients lung transplantation candidates were able to maintain or even increase their 6-minute walk distance regarding just counseling at home. With advanced lung disease patient, that's a good outcome. It's not that bad if they're not going down with advanced disease going to progress. But we had a lot of limitation in that perspective. First, we just have the 6-minute walk distance that we use as an outcome, which not the best way. And we didn't have as much as supervision and adherence. We didn't know with patients how much exercise they were doing it. So we had to go forward and try to do something else. So regarding all the technology tools to improve, the aim of the study was to maybe test the feasibility of the improved home-based program using technology tools and maybe with some supervision of exercise. So we did was 12 weeks home-based, virtual home-based program with these patients with to check and to see the extent to which the 12 weeks rehab will affect different outcomes. I'm going to go back to the outcomes in the next few slides. And we're looking out on the other outcomes to see if this any improvement is going to stay good or maintained after the 3 months maintenance phase. So we use a prospective sequential study with 3 time points. We recruited pretty much all adult patients that were assessed to be listed in our program, the SHIM. So the outcomes are prior outcomes with exercise capacity with the measure of 6-minute walk test. We use the SPPB for frailty measures, the 5 times sit to stand for the functional length strength. With quality of life, we use the St. George questionnaire. And for feasibility, we're looking at the adherence, the acceptability of it, and part of the cost too, that we look at it for the feasibility of that program. So the intervention, which is the main part of what we did. So the first 3 months, we call it the induction phase, and we had the other phase with the 3 months with the maintenance phase going. So regarding the induction phase, so what we did was the virtual home-based program was 1-by-1 method directly by Microsoft Teams with the therapist directly. So it's 1-by-1. They do it, this training exercise with the therapist directly. We use a phase-out design to a bit to improve the autonomy of the patients and improve the long-term adherence to exercise to these patients, knowing that eventually we're going to have a maintenance phase. So we use that going in. The aerobic exercise, we said to them was going to do that 5 times per week. There was a dependently that we used, but they had some guidance and discussion and all that when they had the face-to-face with the therapist regarding all the session for strengthening. Regarding the maintenance phase, after the first 3 months, maintenance phase was an independent program. So they did the same exercise pretty much, but independently. But we still had some part of supervision. So they had weekly phone calls with the therapist to make sure and what some messages and some communication to make sure if they have any question along the way. So we keep it. So regarding the recruitment in around 4 to 6 month recruitment, we assessed around 53 patients following transplant. We had to exclude some patients. First, main part of it was because they were emergency list patient. So we were expecting these patients to be transplanted before the end of intervention. So we excluded them going in. They had some technology problems for some patients going in. So we offered the study to 24 patients. When 4 patients decided it was a bit too much complicated part of the scheduling for them, they wanted to do exercise by themselves. So I was keeping the standard care that gave him an exercise booklet. And finally, we accepted 20 patients accepted to our study. Running 20 patients, after the initial assessment, going to induction phase, so we lost 6 patients around that time. 3 patients were transplanted before the first assessment, and we had 3 patients that had some medical issues and safety issues that we had to exclude at that part. Regarding maintenance phase, so we lost 9 patients around that time. So we lost around for the assessment, but it's good that many of them had the transplant, which is a good news for 8 of them. So we lost a bit more for the assessment and time to and maintenance phase. So running here the results, I don't want to pass around that to going down. We just want a few points regarding to have a bit more male and female around that here. Many ILD patients, which we have for transplant right now, but we had even a retransplant patients, 2 cystic fibrosis going out as a part. Regarding arterial hypertension, so I want to point out that the patients had some too, many of our patients, that these patients were excluded in the same study that we did previously. So the patients were a bit more maybe sicker with our patients with pulmonary hypertension. 6-week walk test was not that bad compared for start for around 240 meters going in. And we had the SPPB score quite high. SPPB normal score is 12, so we have pretty much as maximally, but it was just a part. I'm going to discuss it with the results later on too afterwards. So the results for the maintenance phase, which is a 12 weeks of induction phase. So first, our primary outcomes was 6-minute walk distance. When difference, we didn't see any statistical difference, but the patient maintained, which is not that bad as compared with these kind of patients. We had 2 outcomes that were statistical significance with that. The first one was the percentage of oxygen during the 6-minute walk test. That shows that the patients progressed the disease. They were a little bit more sicker, so they had more oxygen did the test, even was what we've shown. And the other one, we have statistical significance with a 5 times 10 test that we see a difference here with our outcomes. Regarding the SPPB score, we didn't have statistical significance, but it was quite close. But if we check at the effect size, we still have a median effect size of 0.56, which is not that bad as compared going in, but we have a little bit of standard to have a bit outcome. And regarding quality of life, there's no change of path for the patients. If we analyze a bit differently with the minimal clinically important difference, we've seen with the induction phase that these patients around all the outcomes, majority of patients have to maintain or even increase a bit their outcomes, which is a good way. Like I said, with patients are sicker, we see that with the oxygen and all that. Regarding the maintenance phase, many patients seems to maintain, maybe except maybe the St. George questionnaire about the symptoms, but it seems to maintain what they have. Regarding quite fast about the adherence, so we had very high adherence rate to this session, around 91 and 2% of patients. All like our study exercise and step diary was not that great for maintenance going in. Very acceptable patients, they like it a lot going with agreeing with the patients. And our cost is around 1.2 thousand dollars around that per $20 per patients doing it. And we didn't have to learn any tablets and no pulse oximeter alone. So in conclusion, our studies, I've shown that the 12-week ritual prehabs seems to improve the lower limb strength of these patients, maintain exercise capacity, frailty, and the quality of life of these patients. Around the maintenance phase is not that clear because we lost a lot of patients too at the end, but it seems feasible for patients. It was effective to maintain some outcomes. The intervention was well accepted by patients that like that a lot with high attendance rate, no adverse events. And personally, I've seen them after transplant because I'm doing the post-transplant at the same time. And they said to me directly, they very liked doing that with 1 by 1. They like it a lot. We didn't have any outcomes after transplant. We were not powered for that. It was not the goals too, but I've seen slight tendency that the patients were doing a bit better after transplant, even that going home and all that recuperating. So we didn't have powered that too. It's more my clinical expertise and what I've seen with these patients personally. When I was that with the tendency I have look. And going forward, what we want is maybe to check about the impacts of a little bit as a program like that with post-transplant outcomes going with the further study. And the other point is to decide the optimal duration of the program. We decided that 3 months or 12 weeks based on the systematic review done on exercise intervention and solid organ transplant. But now we've seen a big trend in our program. The waiting time for transplant go very much down. That it passed a few years ago was around 2 to 3 years of waiting. Now it's around few weeks to few months. So we have a big change of a little bit how to match a current waiting time. So we have to think about that, which is optimal time for doing the exercise, which induction or maintenance phase that we have to think that about going forward. So I would like to thank the CDTRP at first funding our study, going out my supervisor of PhD, the co-authors of the study, and the Wilkin company that delivers the exercise intervention to the patients going in. Thank you.
So are you able to turn this into a policy change at, say, hospitals, either your own sort of location or other locations?
In our hospital, my main goal is working for these patients is going in, and I know for a long times that I missed out maybe 10 years ago when I was starting and all that patients are not supervising at home, but I wasn't able to. I was the only physical therapist in the lung transplant program. I had to see patients before assessing him after transplant in the ICU. I didn't have the time to do supervision before. And I missed some patients that were transplanted that were not so good prepared. So it's part of what I decided to do my PhD eventually, to start that and see how it's going to go. My goal is eventually to show that in policy making, to implement that in our program. We didn't have the staff yet doing it as compared with the PT. I have like a 0.5 physiotherapy quoting time with me now, which is a bit better. But we transplanted more patients compared when I started was 30 transplant a year. Well, now we're around 80 a year that we want to transplant. A big switch that we did. And patients are a bit more older, they're sicker too, as going in, they're more frail before. So there's a lot of work of doing it. But my goal eventually is to do as a change that do the other lung transplant programs in Toronto. They have the mandatory for a long time here. They had switched to an hybrid version later on. As it was in the hospital base first, they changed to hybrid with COVID. So there's a lot of changing around time, but my goal is yes to implement that. And I had even that qualitative studies that come eventually too. I did interviews with the patients going forward to add on these aspects of it too. So yeah, my goal is to go there. Yeah.
I do have a question. So I mean, it's very interesting how you showed, I guess the training or the exercises that this involves is that strengthening, right? As also the aerobic exercise, and how I guess the patient get access to, I guess, the training equipment to do this, especially if they're doing it from, I guess, from their homes.
Yeah, we said to the patients but we didn't have the fund that much to pay them like equipment is going in to loan, and we're not have the same tendency. They shouldn't have as much donors to help out. I know I talked with the Rickers in Toronto. They have a lot of funds for donors able to buy some equipment for these patients. For us, it's not that much in Montreal. But not only use the staff at home, I use like canned foods or other weights they have for the weights exercise. We use a bit more the leg exercise with the weights and the body going in. Aerobic exercise, we're mainly asking for the walking, but we didn't let the patients did bike or treadmill if they had at home too at the same time. So we'll let them do it, but use these their amounts. But otherwise, we're just walking with guidance about how to the time of the walk, going to a mall or something like that to help out on what we did.
Sometimes we forget that just body weight can work too. It's a type of resistance training. So we don't necessarily need to have weights, heavy weights at home. And as Nicolas said, so in this case, they had whatever they had at home available, the kinesiologist would use. Otherwise, it would be sit to stand exercise or push-ups exercise against the wall, all using their body weights. So that is also a type of resistance training that you can use.
Thank you.
So thank you.
Thank you, Nicholas.